Hello, everyone. Welcome to this week's Genetics, Bioinformatics, and Systems Biology Colloquium. My name is Hannah Mummy, and I'm a fourth-year graduate student in the Bioinformatics and Systems Biology PhD program. And I'm really excited to introduce our speaker today, Dr. Michael Love from the University of North Carolina at Chapel Hill. So Dr. Love earned his PhD in computational biology from the Freie Universität and Max Planck Institute in Berlin. And he is now an associate professor in the departments of biostatistics and genetics at UNC. And the Love Lab uses statistical models to infer biologically meaningful patterns in high-dimensional data sets and develops open source statistical software for the Bioconductor project. Dr. Love is also, in my opinion, an exceptionally engaged and helpful member of the online bioinformatics community. I personally cannot count the number of times I've searched for advice on a bioinformatics question and to end up finding a BioStars post answered by Dr. Love himself. So it's really a pleasure to have him here today to talk about his recent work in providing a common grammar to analyze, manipulate, and visualize omics data. And with this, I'd like to welcome Dr. Love to start his talk.
Thanks. And thanks everybody for tuning in. I was saying earlier, I would have loved to be there, but it didn't work out with, my wife and I both had a travel week and we were like, well, that doesn't work because we've got two little kids. So, thanks again, thanks for tuning in. I'll start sharing my screen. So, I have these up here, but I've also got the chat below, and I'm happy to take questions throughout the talk. And this is kind of a different, I haven't given this before, so I've been working on this project called Tidyomics with a number of people, actually like an international development team. And so I worked on this content just for today. And it's kind of like one long, well, it's not too long, one workflow that I'm trying to demonstrate, like, what's the power of this stuff that we're developing. And so, you may have tuned in for a genetics talk. And so feel free to like make your lunch or whatever while I'm doing this. And then I'll just show you, so all the content is at this link I just put in the chat. And then like the file I'm working off is this file here. So like you could also go try this code out in your RStudio. And so let me introduce quick, first off the project. So it's an open project, meaning that you can start working on this project if you're interested. It's open source software spanning multiple R packages, and it's truly an international developer team. One of the other leads of the project is in Australia. So we always have like 9 a.m., 9 p.m. calls where we flip a coin who's gonna suffer that time. And it's organized on GitHub. So it's a GitHub organization. And then we've used something called GitHub Projects, which I just learned about last year to organize the work, right? Because we're all over the world and we have all these, projects going on and packages. And I just want to show you really briefly what that looks like, because it might be interesting to you if you're doing some bioinformatics work. So we have a year ago at the summer Bioconductor meeting in Boston. Yeah, we said, hey, we're gonna start this project. It's open to anyone, and we're gonna post a bunch of things that we need to do, like stuff that needs to get done for this project to work. And anyone can come and say they're a member of this project and assign themselves to these things. So I can assign myself to something here. And then, once you get down into it, like, this is one we've been working on very recently. So it's speed up this operation in one of these packages. You get down into it and you can have threaded conversations where, people from different labs are kind of contributing how they think we can solve this problem. And then in the end, these things end up being pull requests or new packages, depending on how it goes. So that's been really, that's been a really fun way to do this. And it's been that way we've been able to involve a lot of people who the other One of the other leads is Stefano Mangiola from Adelaide University, and we were able to involve a lot of people who we, have never met before in person. We also have a Slack channel called Tidiness in BioC, which actually predates this particular project. People were asking, like, how do we combine this idea with this idea? And we have a description of the project at this link. Okay, so now I want to, for those of you who have never heard of like tidy data analysis, I want to introduce that concept. I know that, a lot of students who've had a data science class maybe have heard of tidy data or the package dplyr or like ggplot. Like these are kind of popular data science packages. But I'll just kind of introduce the idea and how we're applying it to genomic data. So, the definition of tidy data is one row per observation and one column per variable. And so, sometimes genomic data is tidy, like I would say a bed file is tidy, but other data is not, and it's more shaped like a matrix, or it's not, you get it across a set of Excel files or something. And so that's not tidy, and it's sometimes hard to work with that type of data. And I think a main motivation for the work here is to try to get especially students and trainees like quickly looking at and exploring data. So it's not that this is going to be the be all and end all, and this will be able to do everything like a Swiss Army knife, but it's really trying to get students very quickly on ramped and looking at data and asking, questions and tying things together. And so the idea is that we have all these different types of data, whether like ranges in the genome or gene expression data or spatial or single cell or cytometry. And we want to allow the analyst to use a common grammar to work with that. And we don't want to reinvent a new grammar. And so we're, using the language that people may have already learned if they took a data science class and used dplyr. So the idea of, grouping on one variable and then, summarizing, or the idea of joining things together. We're trying to use the same words. And then be able to quickly pipe into plots and make potentially complex multi-panel figures natively. And on this left-hand side, the important part is that we don't modify the data. So the data stays in its native format. And so for each of these types of data in R, we have objects that are useful for analyzing that type of data. And we're not going to change that. We're not going to like restructure it, but we're going to make it seem like it's in a different format. So
Sorry.
basically like, yep.
Oh, sorry. I think I need to mute myself.
Okay. So the data stays complex, and potentially the analysis is complex, but the language you use to manipulate the data is going to be, we'll try to make that simple. And then I have just a figure here to emphasize that we've got users. Users are also teaching other users through workshops. We've got people who are going down this path of becoming developers from users. And that's really exciting to see. It's a lot of packages. It's not a single package. And we also try to integrate with the wider community, which is both Bioconductor. So we're like kind of a sub project within Bioconductor, but also we try to integrate with like Seurat and obviously with the other tidy packages from Hadley Wickham's folks. Okay, so that was kind of a quick intro. And now I start by introducing like why is it useful to have these data objects, and why do we want to keep those the way they are? So I'm offering that we're going to have a simplified language for operating with genomic data, but we're not going to modify the original object. So I want to just motivate like what is useful about using an object and a formal class system for representing genomic data. So we often have more information than just a matrix. So if you have like features as rows and samples are columns, there's going to be information about the features, where in the genome those are, or yeah, any kind of metadata about the features you're collecting omics data on. And there's of course going to be extra information about the samples, like what condition the sample is in, what day the sample was processed, things like that. And so you want to have that tightly integrated so you don't make mistakes. And then there's also metadata about other things like the organism, the genome build, which release of which annotation is used is being represented here. And so that's the motivation for having an object. And there are many of these objects in Bioconductor, and the conductor part of the name is that you don't if you come up with a new package, you don't create a new object, but you try very hard to use an existing object. So that allows for integration across packages. Many of the functions in Bioconductor are called, like the programming term for them is endomorphic, meaning that they pass out the same object that they take in. Probably something is modified, some information is added, but you maintain all the information and the structure throughout the different functions. So, one of these types of object is called a summarized experiment, which is basically like an expression matrix with information about the rows, which are say the genes, and information about the columns, which are the samples. So it's basically 3 tables of information. So here, like, this is an example of some information about some genes. So gene 1 to gene 4. Here they have some symbol, some made up data about some 4 samples, which are in 2 conditions and 2 sexes. And then I just create some synthetic count data, say RNA-seq counts for these 4 genes and these 4 samples. And then I build my object with this constructor function. So, you probably normally not building these, but they can be generated through other pipelines. And you end up having this thing, and it has this information that's tied together. But so it's definitely not tidy, right? It is a matrix with metadata attached to it. But I would argue it's a useful thing to have this object rather than 3 separate tables. You can do things like reorder or subset. And so here I'm transposing sample 3 and sample 2, so that those 2 columns are now transposed. And also it transposed with that one operation. It also transposed the metadata. And so that's really important. So you don't make wrong inference by getting the metadata disconnected from the data. And then also, like, it can avoid simple mistakes. So here, if I try to assign some data into a slot of this object, it says, oh, by the way, the columns don't match up because this data set has been reordered and that one has not. So this is, simple stuff, but it's the source of a lot of problems in bioinformatics. And so, having that all taken care of by the backend is very useful. And just to say, sometimes people say, oh, but my data is so massive, it doesn't fit in memory. This also works with backends, with, pretty much all the different types of on-disk representations. You can still have a summarized experiment as the class, and then the data is actually stored in a on-disk backend. Or like there's another thing called a delayed array, where you have the representation, but then the operations are delayed. So that's my argument for, like, this is a good thing. Having these objects is great. And nevertheless, it's hard for new users because there's a lot to learn at first. And so, making this object wasn't so hard up here. I listed the 3 tables. But then what can I do with it? Well, you can ask, what are the slots? And it'll tell you that those are the slots. This is a little confusing. It's not exactly the same. It doesn't look the same as up here, right? Up here I said row data, and then that's not a slot, but it's kind of part of this. And then you can say, well, what can I do with it? What are the methods? And you can ask, like, tell me all the methods. And then there's a lot, there's a lot of things. And some of those are interesting and some of those are not. Like, I don't know, maybe people here didn't know you can take two experiments and bind them together and it'll do the right thing. It'll, also row bind the metadata. But, this is not like extremely obvious, like how to learn and play with these things. So I'd argue that many beginners already know dplyr. And I know that just from having given talks and asked who's used dplyr before. So a lot of students have had like one data science class at some point before they end up in a genomics lab. And so they've encountered this before. And they've also seen like ggplot2 to make plots. So really, and this is a selfish thing. Like, I want to have rotation students. I want to get people exploring data quickly and not just making the plots that are in the software guide, but like making, being able to investigate and make their own type of plots. And so I just, this was a way to, working on this type of project was a way to have this be easier for students that they're just getting started. So many beginners know dplyr, and so you can, do things like say, I just want to filter the samples that are in condition X. Here and elsewhere, you'll see this line of code as Tibble. As Tibble is a thing that takes a table-like thing and makes it something that that can be plotted with ggplot or passed into dplyr. It doesn't actually matter that it's a Tibble. I could have put as data frame. Either of those is fine, but this thing is a Bioconductor object. The first thing is called a capital D data frame, capital D, capital F, and you can't directly pass that in. So that one line of code is you'll see that throughout. Sometimes I have to turn something into a Tibble. So that's very easy and I think intuitive. We're just going to filter down to the samples that have condition X, but you can't automatically do that with Bioconductor objects until Stefano and his team wrote a package called Tidy Summarized Experiment. And so when you load this package and you look at the object, it now looks different. So it's printing differently. The object is identical. Nothing has like, if you save this, it's still a summarized experiment. There's nothing changed. It's just printing differently now. And it prints as if it were tidy. So we had these counts. So now instead of having a 4x4 table, it's printing it as if it were 16 rows. And so one thing that happens when we print it that way is we have to repeat the metadata. So both feature level metadata and sample level metadata is repeated, but now we can look at it as if it was a long table. And that kind of informs the kind of operations we can do with it. So it's called an abstraction because we haven't actually changed the object. We've just changed the way we're looking at it. And the way I like to describe this is it's like an API. So we're saying this is the type of thing you have that you can play with. And you can use your tidyverse verbs on this. And so, that's kind of like the way an API works, where it's like, here's the menu of things you can do, and you order something, ring the bell. We'll do something in the back end, but you don't have to worry about the details. You can use your familiar verbs, and then we'll bring you like the dish, which is like whatever the result is of you having applied some operation to this data. So let me show you some examples. So and this is still a summarized experiment. It still has the 4x4 dimensions. So you can use this filter now. So you can just natively pass it through to a filter operation, and it filters out the samples that were in condition X. So now you can see that now the matrix-like thing is 4x2. And in the table abstraction, it's now an 8-row table. Right, so if we save that, we we can then save that, and now that's just a smaller summarized experiment object. You can pipe operations to other operations. And so I think that really facilitates like exploration. So in this case, filter to these two genes. It has a dot in front of it because it's a special column in the abstraction. So filter to these two genes. I want to plot on the x-axis the condition, on the y-axis the counts, and color and group by sex. And yeah, and split that plot by feature. So I think it helps students quickly make plots, which is very important. You can do something more interesting. So say that there's two groups of genes. So there are 4 genes, and I'm going to put them in 2 groups called group 1 and group 2. So you can take the summarized experiment and group by this gene grouping. We also want to keep the data separate in terms of the condition and the sex, and you can compute on any of the data or metadata. So you could, compute on the count values or any other type of data you have. And so we're making here summaries now across genes within condition and sex on the mean and the standard deviation, and make a plot. So yeah, I think that's pretty quick. And while, this code might not be obvious to someone who hasn't worked with ggplot before, but I would say like the manipulation here is 2 lines. So it's very nice that it's, you're very quickly getting answers, and you're using, if you're familiar with dplyr, you're using familiar words, and you can do interesting and complex stuff. It's not just with gene expression, it also works with single cell. So it works with Seurat objects. It works with this thing called a single cell experiment object. So it's basically a summarized experiment, but with extra bells and whistles. So it, for example, has like special slots for reduced dimensions, like if you do PCA or UMAP or something like that. So I've got an example object here, and you can filter the cells that are in G1 phase and plot the 2 UMAP coordinates and color the cells by the total number of counts of RNA. So like just a few lines of code, and people can be making custom plots. And I like that. I like that you're not restricted to just using like the plot UMAP function, but you can really do, your own kind of stuff. Let's see. Because the single cell objects are obviously much bigger than the bulk gene expression objects. So you've got all, thousands of genes, and then now we've got like many thousands or even, getting up to millions of cells. Usually, this does not include the gene level information, but you can access it. So usually, when you're working with these single cell experiment objects, the way that Stefano has written this, it's usually basically doing like cell-based operations. But you can, of course, access and compute on the expression values. You use a function called join features, and then it will bring in the individual gene level expression. But often you're using other functions, say like projecting the cells into an expression manifold or something. You're using some other functions, and then you're again back to operate on them like cells. So I just wanted to show you that it's possible to bring in the gene level expression data, but for speed purposes, it's not always represented. Whereas that's in contrast to the bulk gene expression object, which it was all there, right? So like for every gene, I had a count value in this abstraction. I think I've got one more single cell example. So you can, use any of the like tidyverse operations. So there are functions to help you extract. So I don't know if I had did I show it above? So there's a column here which has called file, which has, kind of a grubby looking information. And I know that the sample is in here. So like there's some data and then something, and then there's an alpha numeric thing, and then there's more. And I just want to pull out the sample so I can use this extract function, and then that tells me the sample. And there's some nice convenience functions, like say you wanted to do a pseudobulk analysis. So aggregate the cells by sample. That's very easy, and it actually turns you back to a summarized experiment now, because this is more like a bulk data set. I'm going to pause for a second and see if people have questions before I move on. The next bit will be more about genomics, like looking at genomic regions. If people want to go
Looks
straight
like
to
no questions, Mike.
Yeah.
Yeah.
That's fine. And again, if you think of one later, you can shoot it in the chat.
I actually have a question.
There
Sure.
we go. Go ahead.
Yeah, so I have used Seurat a lot, and I know that a lot of their plotting functions do return a ggplot object that you can then modify further.
Mhm.
But I guess, can you kind of compare them a bit more? Is it still, you think this is just still easier to modify and work with?
No. I mean, like if you have right, so if Seurat makes a ggplot and then you want to tweak it, I think that's fine. I don't know what the like compelling use case would be here. I think it's, if there was something, if you want to go like off script. So if there's something you want to do that is not clearly accomplishable in Seurat, like I have genes in different pathways, and like per pathway, I want to make some custom computation. That's probably going to the code will look nicer to look at if you did that with this tidy single cell experiment or tidy Seurat. He's also written tidy Seurat. But if you're doing the basic workflow, I'm not going to say like switch over. I don't think it's really like if you're doing something that's a little more complicated, or like the basic workflow isn't doing it for you, you want to make a different plot, or you want to filter the data in a different way, I think these pieces are the way to go.
Sounds good. Thank you.
So I think actually I might have better examples in the genomic part. So where we're working with genomic ranges that kind of give an example, like what does it mean to like do something a little more complicated? And so now this is working with ranges, which like think about like a bed file. You have a bed file. And so you maybe want to ask some questions about where those ranges are in the genome, whether they overlap with other ranges, that kind of question. So we're not talking so much about having multiple samples, we're just focused on, a region based analysis. The first important concept is that, I should say this is going to be talking about a package called Ply Ranges. This was developed by a team in Melbourne. So Stuart Lee, Michael Lawrence, and Dai Cook created this package. And then now it's part of the Tidyomics project because it's very consistent in its goals. In R, we represent like bed file like data with this thing called a G ranges, the genomic ranges, and it has the same information. So just to be a little bit more generic, instead of saying chromosome, they call it sequence. Like it's not necessarily a chromosome. So sec names, it has the range and the strand, and then it can have additional metadata, which is on the other side of this vertical bar. And Ply Ranges has a bunch of functions, convenience functions for reading this in. So like read bed, read narrow peak, etc. So let's say we have a set of 40 ranges. These are just synthetic, obviously. We have 40 regions on chromosome 1, and these have some score value associated with them. And then I have another set of ranges also on chromosome 1, and these are larger. So these are 200 base pairs, and these have a name, so ABC. And so the way that PlyRanges reformulates an overlap question, so you might want to know which of the regions in X fall in these different 200 base pair regions in Y. It formulates that as a join, and a join you can think of as like you have two databases and you're trying to kind of see where they match. And so in normal database language, a join is when a column has the same value, the two columns from the two tables have the same value, but here the identity will be from a genomic overlap perspective. So it could be like a join if this region and this region are directly overlapping, or we could have, you know, more sophisticated things like they have to be a megabase away, or this one has to fall within this other one. But the idea is now to combine tables of information as a join. And so if I pipe X into the command join by overlap, and this is going to be an inner join, I get back X, but I have added on a column from Y. So now X has been annotated with information from Y based on which of these three regions that the range in X falls into. Inner join refers to the fact that there's, if there were ranges in X that did not overlap anything in Y, those have been removed. You can also perform a left join, which retains ranges in X and gives them an NA value because they don't overlap anything in Y, so the value for ID would be NA. And there's many options here. These functions can be, you can have a suffix, whether you want it to be a directed overlap, so strand aware, whether the feature has to fall within or any type of overlap. They can have a gap, and they can have a specified minimum number of base pairs to call it an overlap. So that's useful. And then you can chain operations. So you can say, okay, I want to get rid of the things that have a low score. I'll do the join. I'm going to group this analysis by the ID. So of the three ranges in Y, the ABC, I'm going to group by that. And I want to compute on the score and also count. So now I have average score per range in Y from these X ranges. And I've also tabulated the count of those. And then you can also quickly like pipe the data directly into a plot. So again, filtering and joining, and now I'll just plot all the data. So I've got, these are the scores from the ranges in X grouped by the overlapping range in Y. And again, like just to hit on this point, the verbs are like, no one has had to learn Bioconductor specific language here. These are the verbs that someone would be familiar with if they knew dplyr and ggplot. There's a bunch of convenience functions in PlyRanges package. So you can do anchoring, which is basically like fixing one of the points. So you can fix the 5' or fix the start. The start is the leftmost position, so it's not 5' because it doesn't care about strand, or the end or the center. And then you can modify the range. So say fix the 5' end and make it a 2 base pair range. And then there's a bunch of other convenience functions like go get the nearest range in Y. Or in this case, I swapped Y and X. And you can add also like the distance, where if they're overlapping, the distance would be 0. So that's the quick intro to working with ranges using this tidy language. And then the last thing I wanted to show you was some packages that we developed, or one package that we developed in the lab and in collaboration with another lab at UNC, that kind of assists in these genomic range operations. And so I just have two quick examples, and then I'll wrap up and take questions. So obviously here, one thing we're really interested in this project is modularity. So you don't try to do everything. You don't try to wrap everything up in one function that will have a billion arguments, but you try to let users combine pieces like Lego bricks to make complicated analyses work. And so with that in mind, we wanted to develop a package that is modular in that it doesn't do genomic enrichment analysis, but it just helps you create sets of ranges to enable different comparisons. And so two types of things that people often want to do is create like shuffled data in the genome, or create a matching set. So you might have these regions of interest, say like SNPs associated with some disease or like loci, and this other set, but they're somehow like they're incomparable. Maybe the GC content is very different, or the gene density is very different. And so you might want to have a matching subset of a larger pool of data to make a more equal comparison. So I'll just quickly show what that looks like. These obviously have a lot of options here. And these don't do the comparison itself. You would go back and do that manually using some apply ranges code. So we created a within this package called null ranges, and you can click there to get to the package homepage. We have one branch of functionality for bootstrapping, and here we do what's called block bootstrapping. That was proposed in a paper. I mean, probably more than one, but one paper that we read and that was influential for us was from Peter Bickel at Berkeley, showing that it's very important to use blocks rather than permuting features, right? So if you just permute each feature to give it a new start position, you break down the natural clumpiness of genomic data. And so one thing is like block bootstrapping. And so here, these little trapezoidal things is like I took a piece of the original data and then I moved it over here like a tile. And then I went and subsampled again. And so it's bootstrapping because you can resample the same features twice. Like this set of ranges goes here and here. And the other thing is that Bickel describes in this 2010 paper is you also probably want to do this by drawing blocks from like segments of the genome. So if you're in a high gene density region, you might want to draw from the entire genome, but only populate with ranges from other gene dense regions. And so that's an option here. I won't talk about it. I won't like show you the code to do that, but we provide some segmentations of the genome based on gene density to facilitate that. So it's pretty easy to do this. I'm not getting into the details. For genomic data, the blocks that we found to be effective, and also Bickel, are like 100 KB, that size. So much bigger than this simple example. But you just say, here's my data, please block bootstrap it. In this case, I'm just generating 20 bootstrap iterations, and then you pipe it into another join call. And so we can take the bootstrap version of X and join with Y. And now we have to keep track of two things. We have to keep track of which bootstrap we're looking at and the ID from Y. And then you can say like, how many overlaps were there per element in Y in this bootstrap version of X? And you can pipe that directly into a plot. And we can then compare what we saw in the original data with our bootstrap version of the data. So what I like about this is that it's very customizable. So like there are many details to this analysis, and you can tweak this, to your heart's desire. So the other branch of functionality we created was to help create matching ranges. So like here, the idea is if you have a set of ranges here at the bottom, these have some covariate associated with them. Could be, like I said, GC content, or whether it's whether it's a looped range, any kind of covariate you could imagine. If you want to control for that, so to not have that influence the enrichment, and you have to be careful about what you control for, because if you control for something that's like downstream of the effect you want to measure, then it will go away. But if there's some confounding variation that you want to control for, you can say, here's a pool of elements, and please select from this pool a subset that look like this focal set. And so here I'm creating like an artificial example where like I take X and I subset those that have a high score, and then I just add a little bit of noise, and then I'm showing like I can draw from X to find a subset that looks like this high score set of ranges. And so the way that this looks is the pool is represented by this green density, and we pick from the green density a set that's called the match set, so the blue dashed line that looks similar to the focal set. So the focal set has these high scores, and we're going to subset from the green density a set of ranges that look like the focal set. And so then you can put those together into one set of ranges. So here I'm putting them all together. So now I have a column which tells me these ranges come from this set, these ranges come from this set, these ranges come from this set. So it's tidy in that it's not 3 objects, but it's one long object with a column that tells me the origin. And you can quickly like make comparisons. So here, ask across these 3 sets, what was the average score of ranges that overlapped with Y? Or you could likewise, pipe it directly into a plot. So, it's more lines of code, probably. It could be more lines of code, but I think it's readable lines of code, at least if someone already knows, dplyr and ggplot. So those two, if you want to read more about those, they're published as application notes last year. So wrapping up, I'll say we've implemented like this for matrix shaped objects, for ranges, for chromatin interactions. That's very recently. That was just done by some volunteer from the community for cytometry data and spatial. And if you're interested, you can talk to us on the Slack or sign yourself up on, yeah. So the GitHub is just Tidyomics. And you can, ping us on Slack or email us, and we're happy to take newcomers. And some of the challenges, like these issues, have been described as like good for a new user, if you're kind of new to our development. So I do want to list some of the limitations. So, I still write package code using base R. And one reason for that is something called non-standard evaluation, which is what this all relies on, something called non-standard evaluation. That makes it a little tricky for a developer. You have to, I won't go into the details, but I still write package code with base R. So then that's, now I'm saying that if you bring someone into a lab and they learn this, then they have to go back and learn another language. But I think that's just a natural part. Like, there is no one language. You should learn many languages and understand the interrelation to them. And then the other thing I want to mention is, it's not necessarily optimized for everything. So like basic matrix operations, which are implements in very fast Fortran code. Like, so when you say like calculate the variance of every row, that is then called by a subroutine to some very fast operation that was probably written in like 1970 in Fortran. And so those are just going to be faster using base R. And to show that, I make a data set that has, say, 1,000 genes on 100 samples. And right, so we can do this. We can calculate the row mean in this tidy way, group by gene, compute the average count, and that gives us this table here. But if you compare to base R, which says get the matrix and compute the row mean, it's like 20 times faster. So if you're doing some exploratory analysis, I don't think there's a big problem with doing it the way I showed you today. I mean, it's still fast, like that was 3/100ths of a second. But if you're doing this in a package and it should be very fast, or you're going to do this a million times, or you have, many more, the dimensionality is very high. It's obviously not as fast as like just calling the row mean function. Yeah, and then I just have some like evangelist stuff about please always look at your data, look at PCA plots, look at densities, look at your positive features by eye. There's some more complicated stuff at this. I've been working on this online book. You can check that out. And then I'll finish with the contributors. So Stefano Mangiola is the other big project lead. And the folks who developed the null ranges package is Eric Wanson and Doug. Apply ranges was developed by Stuart and Michael and Di. And then there's all these folks who are contributing on the GitHub, and which is fantastic. And so please reach out to us if you're interested. And the funding is from a Chan Zuckerberg Award and a Wellcome Trust.
Great, thank you so much. We can all virtual applause. So we have about 10 minutes for questions. If anyone has them, they are welcome to unmute or put them in the chat and I can read them out. I can also start off with some of my own questions.
Sure.
So you mentioned more complex applications of these tools. I was just curious if you had an example or anything that you thought was really like the most exciting application or that really pushed boundaries and kind of what that looked like.
Sure. I mean, interactions are really interesting. So like you've got an enhancer and a promoter, and oftentimes people want to do, talk about genome-wide, what happened. Maybe let's say like you're doing a perturbation experiment where you're perturbing enhancers and you want to know how did that perturbation affect genes? What does this tell us about the enhancers? What does this tell us about the genes? There's just a lot of things up in the air. So having, being able to manipulate that with this like tidy data style, I think is going to make cleaner code. Rather than having this all as like separate tables and you're trying to keep track of everything, and you're probably generating a lot of intermediate tables, doing like, let's say you want to like, what do these class of enhancers, when we perturb them in this cell type, how do they affect these classes of genes? That's going to be a really complicated analysis with like pages and pages of code and lots of intermediate tables where you've summarized things. And it's not that that's going to be like really, really easy and the tidy way, but I think it's going to be more legible and there'll be less intermediate tables created. Another example that you can do is you can do nesting. So nesting is when you break apart the object you have into smaller bits and you embed them as elements of a table. And so like you can, like, let's say you have a gene expression matrix, you can chunk that, say by chromosome or by like TAD, and then have an embedded expression matrix in a table. And then you can do parallel operations across those. And so that's a kind of a, that I have an example of that in this tutorial link. It's not it's not super fast, but it's like, yeah, so it kind of depends. You wouldn't want to do this like, you wouldn't want to do nesting where there's like thousands of things you're nesting over, but it, you know, it does clean up the code quite a bit if you have to do something in parallel. So yeah, actually, if anybody's interested in that, you feel free to just ping me and I can give you some pointers.
Great. Yeah, definitely interesting. I work a lot with links information, and it definitely is a struggle to get everything in a common format and then kind of figure out how to work with it. I have another question. Again, if anyone else has any, you can put them in the chat or unmute. I think there's a lot of, as we mentioned, a lot of bioinformatics students on this call, many of whom are kind of working on developing their own tools. And I know you have a lot of experience with this. So I was wondering if you had some advice for students kind of in the development world.
Yeah, I do. So what I've found to be the most relevant thing for development of methods and tools is find a good match, find someone who has a use case, a compelling use case, and basically like use them, you know, use them as your sounding board, because sometimes it's very hard to conceive of like, what do people need? Like, what's the limiting factor for going forward? And we have one idea, like in single cell, I feel like there's a whole period where people are like, oh, the problem is all these zeros. So we're going to like spend years working on getting rid of the zeros. And then the technologists are like, well, how about UMI deduplication? So I just think being, talking a lot to your biologists, oncologists, epidemiologists, whoever you're working with that has kind of the motivating questions, to make sure that you're solving a compelling use case and not just like writing the next clustering algorithm or something.
Yeah, definitely. That makes a lot of sense. Okay, there's a question in the chat from Carl. Is there a benchmark documentation on CPU and memory requirements for various Tidyomics packages?
Yeah. So, one thing is we don't re-implement the algorithm. So like in ply ranges, when we do an overlap, it's actually using the same fast implementation of overlaps that you would get with a normal overlap call. So for, like if you want to know, is ply ranges fast, it's going to be the same speed as genomic ranges. We do have some benchmarking in the manuscript, but I think it's also like, it's more a question of, are you doing an exploratory analysis or are you writing software code? So I'm still going to keep using, the efficient, vectorized, and oftentimes subroutines in R if I'm writing package code. So like I'm going to use, a matrix inverse or like any row-wise or column-wise operation. If I'm doing that in package code, I'm going to do that with the original functions. But so I see this as more like you're doing something custom. You just want to quickly get in and explore the data. So it's not that we're trying to, we are trying to speed some things up. Like there was a group by operation that was slow, I showed you before. And so like, we just found a way to make it much faster. But it's not that our final goal is to like be as fast as the the other implementations. Yeah, but for some demonstration ones, we were comparable to the other implementations if you check the manuscript.
Right. Yeah, that's definitely a problem, especially with single cell analysis. Sometimes we start running things and they take days.
Yeah. Yeah, I mean, I'm not a single cell expert at all. So whenever usually I'm called in, it's already been pseudobulked and somebody wants to do a multi-sample comparison, but like actually working on the single cell data as cells, I don't do that much. And yeah, I think this is useful really for quick plotting of single cell data.
Yeah, definitely.
Other software as efficient as R. So, so like if let's say you want to like do a QR decomposition of some matrix. So there R and Python are calling BLAST, and so it doesn't matter. And MATLAB is also going to like much of the large matrix operation, computationally intensive matrix operations, those are not written in R or Python, but they're calling libraries like C or Fortran libraries. So it doesn't really matter if you're writing an algorithm, like you're going to iterate through a FASTQ file and parse the reads or something. You're not going to want to do that in any of these languages. You're going to want to do that in a more low level language that has tight control of memory and things like that. So for me, R is a statistical environment and a plotting environment, and like data munging, but at that level, like the Python R, pandas and dplyr and data table, these like are comparable ways to like munge some data. If you're working with like things that have billions of rows, you're probably not working in memory anyway. So yeah, it really depends on the task. Oop, and at 4 o'clock, which is now, I got to go pick up the kids from school.
Yep, of course, perfect timing. So I'm just gonna resend the link that you posted initially in the chat. So this is the site that Dr. Love went through in his presentation today. So if anyone wants to refer back to that, you can do so. And thanks again for speaking with us. It's been great having you here today.
Thanks, everybody. Have a good day.